Yeah, we were talking about the properties of myocardium. We have already discussed about rhythmicity, automaticity, excitability, and conductivity. And today we will talk about the contractility. Contractility. So, excitation, contraction, coupling. What do you mean by it? So it is nothing but it describes the mechanism by which the axon potential causes myofibrils, that is actin and myosin contractile protein of muscle to contract. In response to excitation, the sliding movement or actin and myosin, that is myofibril, they of the muscle contract and resulted systole of the heart, the muscle contraction. That is excitation. In response to the excitation, the muscle respond and then it contracts. So let us discuss how does it occur. As the axon potential move over the membrane of the cell or cell membrane, as it moves in the cell membrane, as the axon potential moves over the cell membrane, and when it moves over the T-tubule, this is T-tubule, invagination of sarcolemma. This is the T-tubule, and this T-tubule, the voltage gated calcium channels are activated. As the axon potential move over the T-tubule, these voltage gated calcium channels are open. So the calcium that enter into the cell, and as soon as calcium enter into the cell, it cause activation of calcium release channel, and this is ryanodine receptor. We can call it ryanodine receptor also. This is nothing but the calcium release gate. So as soon as calcium that comes from extracellular space, it enters through the voltage gated calcium channel. As the axon potential moves over the membrane in the area of T-tubule, and when the calcium entry occurs, it activates the calcium release channel, that is ryanodine receptor. And then calcium, which is there in the sarcoplasmic reticulum, this calcium that comes out from sarcoplasmic reticulum in the sarcoplasm, because calcium is very rich in sarcoplasmic reticulum. So as soon as they get chance to come up, immediately the calcium comes out in the sarcoplasmic reticulum. So as soon as calcium comes out in the sarcoplasmic reticulum, it binds with the troponin. So troponin is a protein that the binding site, calcium bind with the troponin, and as it bind with the troponin, they cause a conformational change, and the myosin head attached to the actin side, binding side, and there is contraction or the sliding of the actin and myosin filament, and that is contraction. So contraction will occur when calcium binds with the troponin. So here the calcium have to come inside the cardiac cell to stimulate the opening of the or the release of calcium from sarcoplasmic reticulum. And after contraction, when the muscle or the actin and myosin sliding filament or the sliding occurs, muscle contraction occurs, the calcium which has come from the sarcoplasmic reticulum, that calcium have to be moved or pumped back to the sarcoplasmic reticulum. And because it have to go against the concentration gradient, so ATP is required. So calcium is pumped to the sarcoplasmic reticulum, and also calcium is removed to the extracellular fluid. So by the calcium sodium exchanger. So as it exchanges with the sodium, calcium comes out from the cell, and sodium comes in the cell, inside the cell, and this sodium is pumped to the extracellular fluid by sodium potassium ATPase pump. So the muscle is relaxed and again ready for the new uh, ready for the another action potential. The strength of cardiac muscle contraction depends upon extracellular fluid, extracellular calcium ion concentration. And why the cardiac muscle have to depend? The strength of the cardiac muscle have to depend upon the extracellular calcium ion. Because the sarcoplasmic reticulum the sarcoplasmic reticulum of cardiac muscle is not well developed like skeletal muscle. And therefore, the calcium ion storage is less in sarcoplasmic reticulum of cardiac muscle as compared to the skeletal muscle. And even the heartbeat stops in calcium-free solution when heart is muscles in medium or solution where there is no calcium, the heart cannot beat, it is stopped. So what are the solutions that provide the cardiac muscle to overcome this disadvantage? The solution is the transverse tubule or T-tubule of cardiac muscle is 5 times greater than skeletal muscle. Therefore, the volume is about 25 times as greater than skeletal muscle. Skeletal muscle also T-tubules, but the cardiac T-tubules provide a large space for more calcium storage, extracellular calcium, and also because of the mucopolysaccharide present in the T-tubule, which are negatively charged, they bind with the calcium ion and making available calcium in the T-tubule. So as soon as the action potential move over the T-tubule, the calcium rush into the cell and cause contraction of myocardial muscle. Here, so as the calcium this moves in from extracellular to the intracellular and cause muscle contraction. So this T-tubule, it has larger in size and the presence of the mucopolysaccharide, which is negatively charged, they bind with the calcium. They bind with the calcium and contain the large quantity of calcium within the T-tubule. So that as soon as the action potential that moves over the surface of T-tubule and activate the voltage-gated calcium channel, the calcium immediately rush into the cell because of the high concentration of calcium in the T-tubule. And that's make possibilities to overcome the disadvantage of less developed sarcoplasmic reticulum. So that is how the cell responds to the action potential and cause cardiac muscle contraction or the contractility. So extracellular ion also affects the contractility of the myocardial muscle. And so this is one of the properties. After excitation, they respond and calcium comes in, trigger the calcium release, ryanodine receptor, calcium comes out of sarcoplasmic reticulum and cause muscle contraction. Thank you.